Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Complete Guide to Agarose Gel Electrophoresis and Documentation. Preparing agarose gel, step by step. First, weigh 1.0 gram of ultra-pure agarose powder for a 1% gel consistency. Precaution: if you're preparing a smaller-sized gel, you can adjust the concentration. For example, use 0.5 grams for 0.5% gel in 100 milliliters or 1.5 grams for 1.5% gel, depending on the separation needs of your DNA fragments. Agarose mixture. Preparation: you'll need distilled water, 10 xTBE buffer, a measuring cylinder. In the measuring cylinder, measure 10 milliliters of 10 times TBE and pour it into your beaker. Then add 90 milliliters of distilled water to make it a 100 milliliter 1 xTBE solution. Now, add the 1 gram of agarose powder into the solution. Cover the beaker with aluminum foil to prevent contamination. Precaution: do not stir or mix the powder manually. Let it sit untouched. It will dissolve upon heating. Heating the agarose gel. Place the beaker on a heating plate and let it boil. Important: do not stir while heating. Let the agarose dissolve naturally. Check in between using your eyes. If any specks of undissolved agarose remain, continue heating until the solution turns crystal clear. Setting up the gel apparatus. Here's the complete gel apparatus setup: buffer tank with lid, voltage and timer system, gel casting tray, combs to create wells for samples. Each comb has 15 wells. Use one comb if running PCR samples. Insert the comb carefully into one of the ridges of the gel tray. Pouring the gel with Ethidium Bromide. Ensure the gel is warm, but not too hot. Tip: if your fingers can tolerate the heat for 5 seconds, it's safe to add 16 microliters of Ethidium Bromide. Precaution: always wear gloves when handling Ethidium Bromide. It is carcinogenic. Pour the gel slowly from a corner to avoid forming air bubbles. Let it solidify at room temperature. Removing combs and transferring gel. Once solidified, gently remove the combs by holding both ends and lifting straight up. Ensure the wells are intact. Now remove the side clips to release the gel and prepare for placement into the buffer tank. Preparing running buffer solution. To make the running buffer, calculate the volume according to your buffer tank size. For example, to fill a tank that requires 300 milliliters, mix 30 milliliters of 10 xTBE with 270 milliliters distilled water to get 1 xTBE. Place the gel inside the buffer tank, ensuring the wells face the black cathode side. Note: DNA is negatively charged due to phosphate groups and will migrate towards the positive, red, anode. Sample loading. Tip: place a dark background or light source beneath the gel to clearly see the wells. Start with the 1 kilobase DNA ladder. Load 4 microliters into the first well. Now, load your PCR amplicons, between 3 to 8 microliters, into the subsequent wells. Loading extracted DNA samples. Stick a piece of tape on your lab bench. Add 2 microliters of DNA loading dye onto the tape, 1 dot for each sample. For DNA extracted samples, take 4 to 8 microliters of DNA sample. Mix it gently with the dye on the tape using a pipette. Then load the mixture into the respective gel wells. Starting the gel run. Close the lid of the buffer tank. Set the voltage at 110 volts. If using a DNA ladder, run for 50 minutes. Without ladder, 30 minutes may suffice. Set the current at 450 milliamps. Then press Start. You'll see bubbles forming at the cathode, indicating the current is running. DNA migrates from negative to positive terminal, and shorter DNA fragments move faster, allowing separation by size. Visualizing the gel. After the run completes, turn off the power and remove the lid. You can't view results by eye. Place the gel in the gel documentation system. Precaution: wear gloves while handling the gel. Open the application software and enter the darkroom mode. Choose UV 302 and click Auto Exposure to adjust lighting automatically. There are additional light modes. UV 365 for long-wave UV, blue light, for safer visualization of non-Ethidium dyes, white light, for visualizing protein gels or colored ladders. Final results. Here are our final results showing PCR product bands, DNA ladder, DNA extraction product bands.
If you want to learn how to extract DNA, click the box on your left. And if you're interested in more molecular biology techniques, tap the playlist on your right. Don't forget to like, subscribe, and hit the bell icon to stay updated with my latest science tutorials. Thanks for watching.